Hello and welcome. So today we are going to continue the morphology of bacteria. So this will be the outline and this will be our learning objectives. So as we said in the previous video, we said prokaryotes include bacteria and archaea, and they are basically single cell organisms. So we say they are unicellular in nature and they form part of the earliest forms of life on Earth. And they are able to thrive in various types of environment, including even extreme habitats, hot springs, swamps, and also even in the guts of animals. And so when we say bacteria, it's from the Greek word bacterion, which means the very small stuff. And like we said earlier, unicellular, microscopic, they may be free living or parasitic, and they can perform essential life functions, and their chromosome is made of DNA or RNA, but they lack chlorophyll. Though they lack chlorophyll or chloroplast, some bacteria are photosynthetic autotrophs in nature, and so they perform certain photosynthetic activities. So, like we said, they are ubiquitous, and so they are found almost everywhere, in every niche on Earth. They are found in water, in food, soil, air, and so on. And they can even survive in extreme conditions such as very high pH or very low pH, whether in oxygen or without oxygen, and also very hot temperate regions or not. And their cell wall is structured such that it makes them unique, and the uniqueness of their structure accounts for their pathogenicity, that's their ability to cause disease in man. And so when we pick the bacteria, we see or we learned that they have no nucleus or they don't have any membrane bound organelles, and their DNA is single circular chromosome. They have no histones associated with it. And when we say histones, we mean the protein that provides the structural support for the chromosome. As we may know, each chromosome contains long nucleus of DNA, which must fit into the cell nucleus. And so to do that, the DNA wraps around a protein called histone, which gives the chromosome, more compact shape, as we find. But when we pick the bacteria, they do not have any histones associated with their DNA. And like we said earlier on, their cell wall is made up of peptidoglycan, which is made up of the polymer of N-acetyl glucosamine and also the N-acetyl muramic acid. The N-acetyl glucosamine, what it does is it facilitates the cross-linking between the peptide chains of the peptidoglycan, whereas the N-acetyl muramic acid provides the structural support to the peptidoglycan layer. And also we said they have a smaller ribosome unit, which is a 70S, and they reproduce by binary fission. And so it's one of the very smallest microorganisms that we can describe and can only be visible under the microscope, and we measure the size in micrometer. And so the bacteria of medical importance has a size of about 0.2 to 1.5 micrometer and about 3 to 4 micrometer in length, and can only be visualized under a very high power magnification of about 1000%, by which or with which we are able to view the internal structures of the bacteria. We can classify bacteria based on the morphology, the biochemical or metabolic differences, And even we can also use the immunologic characteristics as well as the genetic characteristics of the bacteria to classify them. And so by the shape or the morphology of the bacteria, we have what we call cocci, which means oval or spherical in shape. We have the bacilli, which means rod-like in shape. We have the vibros, which is common in shape. The spirilla, which is rigid spiral forms of the bacteria. Then we also have spirochetes, which is rather a flexible spiral form of the bacteria. Then the others may be the actinomycetes, which are basically branching filamentous bacteria based on their structure. And also we have the mycoplasma, which basically lacks a cell wall. Now these are some of the shapes of the bacteria. We have the cocci, the bacilli, and some other examples. I think they are better seen over here. And so still on the shapes, we have the A, which is representing the bacillus, which is, if it is only one, plural is bacilli. Then the B is representing what we call coccus. So when the bacteria is oval in shape, we say it is coccus. That is if it is single. But if they are plural, we say it is cocci. And if the cocci are found in strips, or if they are found in chains, we call it streptococci. So streptococcus means they are found in chains or they are in strip. And if the cocci or the oval shape bacteria are found together forming a cluster, we call it staphylococcus. So staphylococus means in cluster. So staphylococcus meaning oval shape bacteria which are found in clusters. Then we may have a single cocci, which is coccus. If there are two, we call it diplococcus. Then we also have the spirillum, which is the E. Then if it is plural, we say spirilla. An example of it is the spirochete. Then we have the comma-shaped bacteria, which are basically vibro. We are going to look at examples of all of these forms of bacteria. And so on this slide, you have the coccus, which is a single oval-shaped bacteria. There are two called diplococci. They are found in clusters. We call it staphylococci. If they are found in strips or in chains, we call it streptococci. Then we have the sarcinae and the tetrad. Tetrad basically means they are 4 in number. So we pick the cocci, and these are small spherical or oval-shaped bacteria, and from the Greek word kokos, which means berry. And so this is almost the same thing as we saw in the previous slide. Then we have the diplococci. Diplo means double, and so they are found in pairs. So examples of diplococci, we have the meningococcus. Meningo from the word meninges, and so the type of bacteria which is oval in shape, which causes inflammation of the meninges, that's the covering of the brain. And so basically meningococcus is the causative agent of epidemic cerebrospinal meningitis. Other forms of diplococci which we must know is the gonococcus. Gonococcus from gonorrhea, and so it is the causative agent for gonorrhea. Then we have pneumococcus, so pneumococcus, causative agent for pneumonia. Then we have the streptococcus in chains, and so these bacteria, they divide in one plane and remain attached to form chains. An example is the streptococcus lactis. We shall look at various examples and diseases that they may cause. Then we have the tetracoccus, which rarely produce diseases in man. An example is the Gafica terragena. Here we have staphylococcus. Like we explained, staphyl means cluster of grapes, and so these cells produce bunches of cocci resembling that of cluster of grapes. That's why we call it staphylococcus. And they, some species cause disease in humans and others in animals. And so the most common ones we have in humans, we have Staphylococcus aureus as well as Staphylococcus albus. Then the sarcinae, sarcinae from Latin word sarcio, which means to tie. And so these cells divide in three planes at right angles to one another in regular pattern. And they are frequently found in air. Their virulent species have not yet been encountered. An example of it is the Micrococcus tetragerna. Then we have the bacilli. Now we are done with the coccus, which are the oval-shaped bacteria. Now we have the bacilli, which are the rod-shaped bacteria. And so example is the Bacillus anthracis, which causes anthrax, the disease anthrax. So as we can see, we have some of the bacilli having the same length as well as the width. Such forms of the bacilli are called cocobacilli. Cocobacilli. And so if it's just a single rod-shaped bacteria, we call it the bacillus. If they are plural, you say bacilli. If the shape, the length and the width are the same, we call it cocobacillus. If they are found in pairs, we call it diplobacillus, just like we had diplococcus. And if they are found in chains, we call it streptobacillus. And if they are found arranged like bricks or blocks, we call it palisades. Then the third one being the vibros, which are comma shaped or curved. And the most common example is the Vibrio cholerae, which causes cholera. The spirilla are long rigid rods with several coils or curves, and they have helical shape with a rigid body. And these are examples of it. So actually, Vibrio, spirillum, and spirochetes all fall under forms of spiral bacteria. And so we've explained that the Vibrio cholerae is a causative agent for cholera. We have the spirillum, which causes spirillum, volutans. Then Treponema pallidum as an example of spirochete, which causes syphilis. So I think this is the same slide that we saw before. Then the actinomycetes are the branching filamentous bacteria, and their unique shape is due to the presence of rigid cell wall, example, streptomyces in them. So as you can see in the photograph. In the mycoplasma, they are known to lack, the cell wall, and so they do not have a stable morphology. So they occur as oval or round bodies with interlacing filaments. Now, after knowing the shape of the bacteria, we need to know the structure of the bacteria. And so we group these ones under the appendages, the cell wall, as well as the cytoplasm of the bacteria. And so the appendages of the bacteria, we have the fimbrial, the pili, and the flagella, which we shall see their functions very soon. Then we have the cell wall, which is made up of peptidoglycan as well as a lipopolysaccharides. And so we explained this in our previous slide, and so on our previous video. And so if not watched the previous video, I will entreat you to go and watch it and get the understanding of all these basic terminologies. The cytoplasm rather contains the chromosome and the placid. And so on your right, we have the structure of the prokaryotes, this being that of a bacteria. So we have the cytoplasm, the nucleoid, we have the flagella, which basically is the powerhouse of the bacteria, aids in locomotion of the bacteria. Then we have the ribosomes, as well as some capsules. So let's look at it into detail. Now, when we pick the pili or the fimbra, we have the hair-like structure, which are proteins, and what they do is they help the bacteria to attach itself to nearby surfaces and structures, and they are actually very hard to wash them away. and they also use it. Some of them have specialized sex pillows for conjugation. Then, when we look at the flagella, what we find is that some bacteria have flagella whilst others do not. And so if the bacteria does not have flagella, we call it atrichus. So atrichus means it does not have any flagellum. But if it has flagella, then we must know where the flagellum or the flagella is attached. And so if the flagella is attached to just one end of the bacteria, we say it is monotrichus, as you can see in the in the object A. Call it monotrichus. An example of monotrichus is Vibrio cholerae. And so the causative agent Vibrio cholerae is a monotrichus, meaning it has only one flagella at one side of it. Then we also have the B, which is amphitrichus. So amphitrichus has one flagellum present on each end. So I think I made a mistake with the labeling. So if it has one flagellum present on each end, as you can find in C, we call it amphitrichus. So it has been interchanged. So C is amphitrichus. An example of amphitrichus is alcaligens faecalis. And these alcaligen faecalis causes endocarditis, can also cause meningitis, as well as some skin and soft tissue infections, and even urinary tract infections. Then, if the flagella are present at one or both sides of the bacteria, we call it lophotrichus. An example of lophotrichus is the spirillum, which causes rat bite fever. Then we have the peritrichus, which has numerous flagella present all over the bacterial body. And the example of the peritrichus is Salmonella typhi, which causes typhoid fever. So these are the examples that you need to know. So on this slide, we have the bacteria with its inclusion bodies, as well as the appendages and whatever we have in the cytoplasm, including the flagellum. So you take your time to go through this slide and get to know the functions. Next, we are going to look at the structure itself of the bacteria. And so it's made up of glycocalyx, meaning we have glycocalyx, glyco coming from sugar, and so sugar coats which are tightly bound together, and they form what we call the capsule of the bacteria. And this capsule protects and prevents the bacteria from drying, and also prevents them from being phagocytosed. And they also provide a slimy and like a slimy nature, or a slimy body, or a slimy component of the bacteria, which sometimes makes them actually non-visible by our immune cells, and so they form an important component of biofilm. Now, when we look at the outer membrane of the bacteria, we are able to distinguish between them based on the type of cells that they have. And so we may have the gram-positive bacteria and the gram-negative bacteria. We are going to look at this structure in detail after this slide. But before that, as you can see, both the gram-positive and the gram-negative bacteria share certain things in common, including the proteins, the phospholipid, as well as the periplasmic space and the cell membrane. But if you look closely, you can see that the gram-positive bacteria also has other structures which the gram-negative doesn't have. Example, the teichoic acid and the lipoteichoic acid. And if you look at the structure of the gram-positive, you can see that it has a very thick polysaccharide. It has a very thick outer membrane. And if you compare it to that of the gram-negative, you see it has a thinner outer membrane. However, the gram-negative bacteria also has an extra phospholipid, and this is why we call the gram-negative bacteria as having what a phospholipid bilayer. And we are going to look at what it actually causes. And so this being the outer membrane, found in the gram-negative bacteria. And so these differences between the gram-positive and the gram-negative bacteria actually is very important, and we are going to look at why they are very important. However, the things, some appendages, and the structure itself sometimes are the cause of some of the symptoms that the bacteria infection may present, such as the fever, the shock, and also the diarrhea that we may find in bacterial infections. And so I think we've talked about this already, but we have certain antibiotics which work by inhibiting the cell wall of the bacteria. Others also work by inhibiting the synthesis of protein in the bacteria. And so based on how the bacteria structure is, we have certain chemicals which we also use to prevent them by way of treatment. So this slide is showing us the bacterial cell wall having the peptidoglycan chains which are joined together by the teichoic acid. And this is basically found in the gram-positive bacteria. And so the gram-positive and gram-negative bacteria have differences in the cell wall, as we've seen. And this explains why we have differences in treatment outcomes, as well as our ability or the pathogenicity of each of these groups of bacteria. And so from what we just saw, it is important for us to note that the gram-negatives are less sensitive to penicillins as a result of their thinner cell wall and the outer membrane. And also the gram-negative cell envelope are much more complicated than the gram-positive bacteria. The complication arises from the fact that they have the phospholipid bilayer, whilst the gram-positives have only one phospholipid layer. And so if you look inside the bacterial cytoplasm, you have the nucleoid, which is the area that contains the bacterial chromosome, be it the DNA or the RNA. And inside the cytoplasm as well, we have some other non-membrane bound organelles such as the ribosomes, and this is going to be a 70S ribosomes. And also we have certain antibiotics which work by inhibiting the synthesis of proteins, as I've mentioned before. Now, when we look at the bacteria and we look at the inclusions, some may include the granules of sugar, sometimes lipid storage, and so on. But we have certain bacteria which are very difficult to destroy or which are very difficult to kill, as we saw in our introduction. And those bacteria are the ones we call the bacteria endospores. An example, we have the bacillus and the clostridium. They are the hardest of bacteria, and it's very, very difficult to kill with heat or chemical. And so with these kind of bacteria, we are able to destroy them using the autoclave at 121 degrees Celsius under pressure to penetrate the thick coat of the bacteria and destroy the genome of the bacteria. And so it is only by use of the autoclave, the process which we call sterilization, that we use to destroy the bacteria endospore. And as we've seen, before they reproduce by binary fission. And with the binary fission, as we know already, we have the parent cell, and before it divides, we have duplication of the chromosome. And once the chromosome duplicates, then the cytoplasm also divide. And so at the end of the day, we are going to have two separate daughter cells which share very common similarities with the parent cell. They have almost everything. Thank you very much for your time.